We are looking specifically at the cell membrane today, and it is asking us to describe the structure and function of the cell membrane based on some specifics there. Right, so we've seen how the basic cell is structured and that it may contain heaps of organelles. This lesson focused solely on the structure of the cell membrane, and the membrane is a highly specialized organelle, I guess you could call it, to contain all of the internal contents of the cell, but strategically it has to allow substances in and out where it's needed. Now, many scientists over the 20th century had a crack at determining how the cell membrane was actually arranged without the, help of really good microscopes. So Gorder and Brendel determined in 1925 that it was made of a fat molecule known as a phospholipid. And they decided to measure the number of phospholipids that were in a red blood cell. When they compared the total to the surface area of the cell and the known size of a phospholipid, they came up with twice as many phospholipids. So they decided that there must actually be a double layer of phospholipids, and they drew it like this. Now, Davson and Daniele came up with a similar picture. They actually had a photograph there in 1935, and they looked at two adjoining cells. And what they saw looked like three really clear layers. We know that it's actually back-to-back membranes on two different cells, but they proposed that the cell membrane looked something like a sandwich, right? It had something on top, something in the middle, and something in the bottom there. Bread on the outside, something else in the middle. And they knew that there were phospholipids involved because that had already been found out, but they knew there was also proteins, but they couldn't quite figure out how to make that a picture. And so this is what they came up with. And there's quite a few problems with it, not least of them being that they had no idea how certain molecules would move in and out of the cell in this kind of sandwich-like fortress. So after some playing around with this, this model was basically debunked, and Singer and Nicholson emerged in 1972 with the most likely model. And this model showed that the bilayer of phospholipids with proteins were embedded in the bilayer. So it had all the right elements, just in a different configuration. And they called this the fluid mosaic model, okay? And seeing as though it was pretty fluid, it moved really well, it was able to change shape and move with the cell, but it's still being made up of those components all sitting side by side. Now, this is the model of the cell membrane that we can confirm and hence we use today, and you'll see a lot of these kinds of pictures. Now, the cell membrane encloses the entire cell. It separates the internal environment of the cell from the external environment. In a single-celled organism, that's just the outside world. Otherwise, in a fused multicellular organism, it's extracellular. So it's made up of many components, and they make up essentially a fluid carpet and allows for the cell to have things coming and going when it needs to, which is what we call selective permeability. It allows some things and not others, depending on the environment and the requirements of the cell at that time. So a fluid mosaic model can be depicted in many different ways, but always remember that this is a 3D structure. It is not a 2D piece of paper. So the bilayer of phospholipids and then proteins acting as kind of channels for things to come and go. It's very fluid, it moves, the phospholipid bilayer is moving, and there's also cholesterol and carbohydrates in there for various reasons. And we call it that fluid mosaic because a mosaic is made up of lots of different pieces all pushed together. The main components that we're going to talk about are a phospholipid bilayer, proteins of different kinds, cholesterol, and carbohydrates. And you'll see even there's four similar but different depictions of this. And they all connect together in that jigsaw as the fluid mosaic here. So we've got our phospholipid bilayer, which are those blue structures there with their little tails, and they have hydrophobic legs, meaning they're afraid of water, essentially. They're non-polar, and they point towards the middle, and the hydrophilic heads will sit on the outside. And they will arrange themselves in this fashion if left alone. They'll just kind of form this because those legs want to hide from the extracellular environment, and the heads are fine with it. Now, there's large proteins embedded into the bilayer. There's transmembrane proteins that go all the way across. There's some that just sit peripherally on top as well, and they're involved in the transport of substances. They're involved in cell-to-cell recognitions, enzyme and hormone signaling, things like that. Glycoproteins are proteins with carbohydrates attached to the top, which are involved in cell-to-cell recognition and adhesion, so they're sticking to one another. And there's cholesterol, those little yellow things in there. They're embedded between the phospholipids, and it actually stabilizes it, so it makes it slightly less fluid so that it doesn't all just fall apart. Now, there's so many depictions of this, and some of them are super simple like this one, and some of them are showing all the components really nicely. But remember, it's that 3D structure. You've got to think, hey, it's like the latex around a water balloon or something. It's not just a flat doona on top. So for the cell to function and perform all the basic requirements of life, it's got to gain nutrients from its surrounds somehow. And the selective permeability, is going to depend on what's needed at the time, what needs to exit if there's wastes that need to leave. So we'll look at lots of different ways in which substances come and go in the next lesson. But the focus of this one is purely structure and function. So understanding all those components and what they do.